All right, thank you. Hi, everyone. My name is Farron Isaacs. I'm a professor in the MCDB department here at Yale, as well as biomedical engineering. I'm here in my capacity as co-founder of Pearl Bio, which I co-founded with a longtime collaborator, Mike Jewett from Stanford. So at Pearl, we have developed a suite of innovations in synthetic biology and genome engineering that we have brought together to basically recode life towards the production of programmable biologics. Oops, where are we? Okay. Multifunctional biologics are really emerging as the future of precision medicines. Examples are shown here, where, for example, ADCs, where you attach drugs to antibodies, serve to really drive targeted killing of cancer cells, or GLP-1s have all been drugged. What's important with all of these is that they've all been modified and functionalized to be endowed with important therapeutic properties. In the case of ADCs, you're attaching drugs to cysteines or lysines. In the case of GLP-1s, you're encoding synthetic amino acids as well as attaching lipids. The multifunctionalization is really important in really endowing important therapeutic properties, programmable pharmacokinetics, tunable DAR, et cetera. The challenge, though, and the problem that we're looking to solve is that the technologies that are conventionally used to endow these tunable therapeutic properties are very difficult to engineer and present challenges for drug approvals. At Pearl and my lab, we've basically been developing innovations in the engineering of genomically recoded organisms, which effectively repurpose codons and allow us to encode multiple distinct synthetic amino acids into proteins. We've been issued the blocking IP on the use of GROs to encode synthetic amino acids into proteins. The way to think about it is that nature has 20 amino acids. We've got 22 and counting. You can read about important advances in this technology in a paper we published a few months ago. Pearl is using this technology to encode these synthetic amino acids to endow the ability to tune some of those key therapeutic properties, like half-life or DAR. We're first pointing this technology to unlock the potential of nanobodies as diagnostics and therapeutics. Why nanobodies? Nanobodies are the smallest antibody, 10% the size of typical full-length antibodies. They've got superior stability as well as ease of biomanufacturing. Distinct from antibodies, they also have high tissue penetrance, but they suffer from short half-life, which has made them a challenge to drug. But paired with the Pearl technology, as you'll see in the next few minutes, we're really developing a capacity to change this paradigm. So if you consider a nanobody and thinking about targeting a tumor microenvironment, you can basically encode synthetic amino acids, and in one capacity, we can attach fluorophores or radiolabels to effectively tag cancer cells to aid in surgery or diagnostics. We can use the same technology, and instead of attaching a fluorophore or radiolabel, attach a cytotoxic payload that allows very targeted delivery of these nanobodies, distinct from antibodies, allowing much better penetrance of tumors and more efficacious cell killing. To give you some examples of how this actually works, I'm showing an example of a multifunctionalized nanobody drug conjugate that only Pearl can make. On the left, what you'll see here is some cell killing data that benchmarks nanobody drug conjugates made by Pearl versus 2 antibody drug conjugates that are on the market. As you can see with the Pearl drugs, 2 of those antibody drug conjugates show improved cell killing versus the antibody drug conjugates on the market. But importantly, we repurpose a second synthetic amino acids to attach lipids that allow us not just to extend, but tune the half-life of these molecules, allowing us to multifunctionalize new classes of nanobody drug conjugates. We've got distinctive blocking IP on the technology behind this that's been developed for over 15 years. We've got a great team who have really pioneered the development of these technologies. Just a quick history. My co-founder, Michael Jewett, as well as my other co-founder, Amy Schwartz. Mike Jewett and I were postdocs in the lab of George Church, who's also part of our team, where all these ideas were first generated over 15 years ago. I'll just close by saying that we've used some seed financing from Coastal Ventures, as well as some non-dilutive funds, paired with some farmer partner deals to launch the company. And we're now seeking a Series A to grow the senior management team and advance these molecules towards the clinic over the next 2.5 years. Thank you.